Good afternoon. I know there's still people coming in, but in the interest of time, I think we'll get going. And thank you all for coming to what is a very special day for the divisions of Pediatric Hematology, Oncology, Blood and Marrow Transplant, and Pediatric Epidemiology. We have had the privilege now of celebrating the legacy of Dr. Mark Nesbit for now this is the 25th anniversary lectureship. And no one more fitting to deliver that lecture than Dr. Bruce Blazar, who completed medical school in 1978, came here, completed all of his pediatric training here, including fellowship and a postdoctorate, and has risen to the level of Regents professor, really focused on bone marrow transplant and is an international leader in the field, and really a testament to the program that Mark built in conjunction with many, many others in the field. And Bruce will walk through that history and legacy, and it's a real pleasure, privilege, and honor to introduce Dr. Bruce Blazar.
Thank you. This is really a special privilege and honor for me to be able to deliver this lectureship. I started in Pediatric Hematology Oncology Fellowship in January of 1982, so I feel like I've been preparing for this for 34 and a half years, even though it's the 25th annual lectureship. So I just have a declaration that I have financial interest in intellectual property in the field of regulatory T cells, and this is managed in accordance with the University of Minnesota Policy and Oversight Committee. So I wanted to start with this picture, which I had provided to me by an anonymous source, but this is Mark and Deb Nesbit, and this is really a team for the ages. This is a 60-year partnership, and I think together they have really led a lot of activities here, both in the division itself as well as a number of charitable organizations. So just a few, for those of you who don't know, Mark started as pediatric here in 1959. He became a full professor within 6 years after completing fellowship, which is probably a record. He was section head from 1979 to 93, associate clinical director of the Masonic Cancer Center from 91 to 96. And he led studies, truly led studies that markedly improved outcomes in pediatric leukemias, both acute lymphocytic and acute myelocytic leukemia. And he formed intergroup studies in pediatric bone cancer, particularly Ewing sarcoma. He published more than 300 papers while he was doing all these activities and trained more than 100 fellows. He served for a mentor and role model for myself and countless other people, both in the University of Minnesota and outside. His leadership roles are numerous: Histiocytosis Association of America Board of Trustees, Outstanding Investigator Award. He was a founding member and vice chair of Children's Cancer Study Group, which is now more than 220 institutions. He was the past president of the American Society of Pediatric Hematology and Oncology and recipient of the Distinguished Career Award. He was the first U.S. president of the International Society of Pediatric Oncology, and he received an Outstanding Achievement Award from the American Cancer Society. If this wasn't enough, I think it's really important to point out that Mark has a dual role, and he had leadership roles in multiple charitable organizations, including Make-A-Wish Foundation of America on the board of directors, Combined Health Appeal of Minnesota as a past vice president, Ronald McDonald Charities, which he was a consultant to the board and special recognition award, and he was one of 6 physicians honored by community caregivers by Minnesota physicians. It's not many people, if any, who can walk these different areas of leading administratively sections, leading international and national studies, being in charge of multiple organizations, and contributing so substantially to charitable organizations, including the Children's Cancer Research Fund, which funds a lot of us. So this is an old picture. This is Mark and Norma, who's here. Diane Arthur, whom you may or may not know, is a cytogeneticist. John Parentis, now at Cincinnati Children's. This is what I used to look like, not anymore, and this is Bill Woods. And this, amongst other people, was a very small but dedicated team in pediatric oncology and bone marrow transplantation that together worked to try to improve outcome of children with cancer. So Mark's notable quotes: "It's tough to fight cancer." Bill Woods used to show this on a picture on his wall. For me, when I was asked to provide my research projects, I gave him 32 ideas. His first response is, "You can't be serious." And then the second response, which I remembered very distinctly, is, "If you're good enough to stay here, you'll figure it out." And 25 years later, I've got some of these completed, but I'm still working on a number of others. So as a general overall schema for hematopoietic stem cell transplantation, you give chemotherapy with or without radiation therapy to try to eliminate tumors and achieve engraftment. There are a variety of stem cell sources that can be used: bone marrow, peripheral blood, and cord blood, as led by John Wagner here. And then there's a process of hematopoietic recovery, immune recovery, and the treatment of the underlying disease. More than 1 million patients have been transplanted as of December 2012 worldwide. There are more than 22 million donors registered, 1,400 transplant centers. There's 7,000 stem cell transplants done each year in the US, with more than 30,000 allogeneic stem cell transplants done worldwide per year. So there are 3 simple concepts to stem cell transplantation. One is non-malignant and malignant disorders of the hematopoietic and immune system can be treated by a well-tolerated conditioning regimen followed by hematopoietic cell rescue. Patients will have rapid recovery of the hematopoietic immune system that's functional and tolerant of the host. And allogeneic hematopoietic stem cell transplantation will provide an ideal platform for tumor reduction and prevention of relapse. So these sound simple, but teams over the years have struggled with this. This, I think, was a truly internationally recognized team of Norman Ramsey, who led pediatric bone marrow transplant, Mark Nesbit, who led the section in hematology oncology, and John Kersey. Mark was squarely in the middle of this and was a catalyst for these studies. So why is this so hard? Well, first of all, chemotherapy and radiation toxicities vary between individuals. They're both genetic and environmental factors. And Les Robeson, who's here, has studied a number of these genetic and environmental factors for chemotherapy. And I think this is one of the foundational principles of why this is difficult, is because there are polymorphisms and individual differences in both metabolizing drugs and responding to those, and environmental factors, including most recently recognized the host microbiome that influences. And there really aren't very good fully predictive formulas for toxicity that exist for many of the chemotherapy regimens. So one of the early support mechanisms from Children's Cancer Research Fund was a busulfan pharmacokinetics lab that Bruce Bostrom, who was a fellow with me, directed. And we had this initial observation that oral busulfan and Cytoxan didn't result in uniform and sustained engraftment in pediatrics patients. If you compare looking at the adults versus children in a paper we did with Louise Grashow, there's a huge variability in the busulfan exposure, ranging over 50%. And children have very low levels of the same drug at the doses per kilo compared to adults. So this led to a series of papers that Bruce Bostrom wrote on busulfan pharmacokinetics in children with AML and in children and adolescents. And this provided some of the foundational reasons to develop an IV busulfan called Busulfex, where now you can have a very linear model and predictive model, so you can pick how much busulfan exposure that you want. And we've now switched many of our protocols to IV busulfan. And this is a result of a divisional supported research activity that Bruce Bostrom led. So the current solutions for this, I mentioned, are reduced conditioning and regimens and pharmacokinetic targeting. What are the future solutions? More selective myelolymphoablative therapy. There are antibodies and drugs that target stem cells and immune cells that do that. There's radiation modifiers. For example, one could transiently inhibit a p53 pathway that's involved in radiation sensitivity. I'll show you information on speeding tissue repair with IL-22, which is a protein, as well as keratinocyte growth factors, which are also proteins that can stimulate tissue repair. And we need better predictive algorithms for chemotherapy, metabolism, radiation repair, the effective age, and the host microenvironment. So this is keratinocyte growth factor. It's a protein produced by mesenchymal cells and epithelial cells. It binds to a receptor that's present on mainly epithelial cells. So in studies led by Angela Paniskalsis-Mortari, one could give keratinocyte growth factor before radiation, try to build up the epithelium so it's resistant to the side effects of graft-versus-host disease, which attacks the epithelium, and the conditioning regimen therapy, which attacks the epithelium. And this resulted in a significant prolongation of survival. And this was the first studies to date that looked at this protein in the context of hematopoietic stem cell transplantation, conditioning, and allogeneic graft-versus-host disease. In a phase 3 randomized study done by Ricardo Spielberger, which we participated in, one could look at the keratinocyte growth factor treated group, palifermin versus placebo, and the severity of mucositis, which was grade 3, grade 4 is the worst, grade 2 is the mildest. You can see that these were very severe in the placebo group, and now they are reduced by the palifermin group, which led to the approval of palifermin for intensive chemotherapy regimens in autologous transplant. We did a randomized study here of 100 patients, placebo versus KGF, and there was a significant reduction in mucositis, oral mucositis, particularly in the radiation therapy group, where there were 54 patients treated here, and this significantly reduced severe oral mucositis and the severity in studies that were done here with Dan Weisdorf and myself. So in terms of rapid hematopoietic cell recovery, full recovery takes weeks to months. It varies between individuals, both qualitatively and quantitatively, despite similar donor grafts. Host factors that can influence this remain incompletely elucidated. Some of them include immune-mediated resistance of the host against the donor graft. The bone marrow can have stromal cell injury, and the stromal cells support hematopoiesis. That can affect stem cell homing, differentiation, and proliferation. The current solutions have included infusing high numbers of stem cells, megadose transplants pioneered by Yair Reisner in Israel, and double cord blood transplants pioneered by John Wagner here, as well as ex vivo expanded stem cells. One can look at improved homing to the bone marrow. There are a variety of ways of doing that. One is to add sugars to the bone marrow so they home to the graft. And then there are cell-based therapies such as mesenchymal stem cells, graft-facilitating cells, and regulatory T cells that have all been shown to improve hematopoiesis, and growth factors that we know can support red cells, white cells, and platelets. So the divisional contribution in this area, I think, is best illustrated by these studies from John Wagner, which he led. And this is a protein that was identified through high-throughput screening by a Novartis scientific research institute. And this is known as STEM-regenin-1. It binds to an aerohydrocarbon receptor ligand. And this is a first-in-human study conducted here, led by John. And what you can see is that the stem cells, denoted by these cell surface markers, expanded 100-fold in culture, and this was unprecedented at the time. And if you give two blood units, one of which is expanded and one of which is not, the expanded unit engrafted significantly better in patients than those who received an unexpanded unit. And the time to hematopoietic recovery was markedly accelerated if you had an expanded unit. So I think one of the approaches in the future is going to be identifying new molecules that in vitro or in vivo can help stem cell expansion in the time of neutropenia and get people out of the hospital more rapidly. And studies from this institution have so far shown the greatest ability to do that through these studies from John. So what are the future solutions to this? We need improved assays to quantify stem cell capacity and homing. We don't know how to track human hematopoiesis very well. There are some new studies that are starting to address this question. If we can track better, we can understand variables that affect reconstitution. We need to identify new molecules, drugs, and approaches for in vivo expansion of hematopoietic stem and progenitor cell graft. And then I think the long-term future might include universal donor embryonic or induced pluripotent stem cells, which can make self-renewable and regulatable hematopoietic stem cells for infusion. And this would allow one to get eventually an off-the-shelf product that would be a continual source of hematopoietic stem cells, so we wouldn't need to harvest people for peripheral blood or bone marrow or use cord blood units, but rather bank these off-the-shelf third-party products. So Troy Lund and Tiffany Glass, who's working with both of us, developed some novel approaches to screen molecules that would promote hematopoiesis. And Troy with Tiffany created a stromal cell-derived growth factor transgenic and transparent zebrafish. So these are zebrafish, which are very small creatures, where you can see when you inject hematopoietic stem cells how fast they home, where they go, and you can bathe them in water containing molecules that can stimulate their homing or engraftment ability. And this is just showing animals that express the stromal cell niches that are denoted by these red areas that are stromal cell-derived growth factor, where the stem cells home. And through a series of papers, they were able to show how useful this model is for that purpose. And Troy's gone on to find new molecules that helps speed hematopoietic recovery. In a second approach, Bo Weber, who was working with myself, Jacob, and Mike Kaiba, sought out to identify during his PhD transcription factors, which are master regulators that drive definitive hematopoiesis, and then develop a novel reporter cell line for high-throughput screening. So hematopoiesis begins in the yolk sac in rodents. It goes to the AGM area, then the fetal liver, and then the adult bone marrow. And it was known that a particular transcription factor goes up very early in the yolk sac, and these are primitive and embryonic hematopoiesis, but it does not work in adults, and so adult mice, and so then this goes down, and this other promoter goes up. And Bo went on to identify the elements in this promoter here that regulated definitive hematopoiesis, and he did this with in vitro and in vivo studies, looked at methylation patterns and epigenetics, and was able to identify that these two regions, in particular parts of these two regions, controlled the ability of the hematopoietic cells to have the ability to reconstitute an adult mouse. And so with that, he developed a reporter line which is driven by this transcription factor so that the cells turn green, and this enhancer so that the cells turn red. And you could follow this by putting it into inducible pluripotent stem cells or embryonic stem cells, and when the cells are both red and green, they have achieved definitive hematopoiesis. So through a grant through Regenerative Medicine Minnesota, which Jacob leads, we have a collaboration with 5 Prime, which is a company in California, to look at a high-throughput screening of an extraordinarily extensive library of 5,800 cell surface and extracellular proteins to look for new candidates that we hope to eventually bring into the clinic. In terms of rapid donor immune recovery, this requires months to years. Patients are highly susceptible to infections and tumor recurrence because you need an immune system to keep infections and tumor recurrence under check. The most vulnerable patients are aged patients, those with high comorbidities, prior extensive chemotherapy, or acute or chronic GVHD, and they recover the slowest. So the field has moved in part to reduced intensity conditioning regimens and to reducing immune-mediated rejection. There are antibodies that one can give to inhibit T-cell or NK cell-mediated rejection, and better supportive care, particularly antimicrobials and antifungals, as well as improved immunosurveillance assays. And to give you a feeling for this, these are data from Juliet Barker, who used to be here at Sloan Kettering now. These are CD4 counts. This is the normal range. Pre-transplant is normal range, and then they stay low, pretty much don't even get to lower level of normal to at least a year. And if you look at the number of infections is inversely related to the number of CD4 cells. So you have to get to counts of 500 or more to get a lower level of infections. And if you look at tumor recurrence or event-free survival, those patients that recover their lymphocytes by day 30 fastest have a higher disease or event-free survival than those who recover slower. So that led to ways of trying to control this. One is to get rid of GVHD, graft-versus-host disease, which is a donor anti-host reaction where the T cells and graft kill the recipients as being foreign, and to look at better diagnosis and predictive algorithms, to improve GVHD prevention with more selective approaches. Post-transplant Cytoxin for 2 days is one of those because it depletes alloreactive T cells and leaves non-alloreactive T cells alone. Cell-based therapies have now included polyfunctional antiviral or antifungal T cells, and suppressor cells or regulatory T cells to eliminate GVHD and spare the patient from immune suppressive drugs. So the future solutions to this, I think, include ways of stimulating tissue repair. I'll mention T progenitor cells, which are thymic precursor cells, and IL-22, which is a cytokine that helps with tissue repair, that one can infuse these cell populations, or gene editing approaches for gene editing, which is to change the genome or to add in a gene where it's defective, and that would allow autologous instead of allogeneic transplant, as well as immune cell therapies for cancer, which have now taken the place in partly of conditioning and hematopoietic stem cell transplantation. So this is just a review of this process. One can take cord blood stem cells and through a series of growth factors expand these cells until they attain a sufficient functional maturity. These cells will then go to the thymus where they mature and they can produce T cells that go into the periphery. And by looking at in vitro approaches to speed this process, we don't ask the recipient to do that. We give them a marked head start by doing it all in vitro. And these are data from Michelle Smith collaborating with myself and Heather Stefanski. And this is looking at given these T progenitor cells, and they go to the thymus, they increase over a period of 3 weeks. They go throughout the body because they're not specifically only to the thymus. And if one looks after the transplant, here are these cells there that are leading to a significantly higher number of donor cells based on giving these progenitor T cells that gives the immune system a marked head start. And this is being developed for an infusional protocol led by Heather and John Wagner to bring this therapy to patients. So a second approach that I mentioned is that you can give growth factors that stimulate the tissue repair mechanism. So we collaborated with Alan Hanash, Marcel Vandenbrink at Sloan Kettering, and this is a diagram from one of their papers. And this is looking at radiation and pre-transplant conditioning, which injures the intestine. There's a pro-inflammatory molecule that increases, and the alloreactive T cells eliminate this cell type called an innate lymphoid cell, which produces IL-22, and this IL-22 stimulates the intestine to repair itself. So what happens is the GVHD causes intestinal injury. It eliminates these protective cell populations present in the intestine, and the net result is a slow tissue repair. This concept has now been brought to clinicaltrials.gov. There's an IL-2 fusion protein that they're going to be testing to see if it can directly stimulate tissue repair. In a different approach, Mike Veneris' lab has developed cell therapies that are involved in innate lymphoid cells type 3, which is what these cells are. He can make these from cord blood. They produce IL-22, shown here, and this might represent a cell therapy approach to complement the IL-22. This actually has certain advantages because IL-22, as with many cytokines, has pro-inflammatory effects, whereas these cells, if they home to the intestine, it's really the local production of cytokines that's helpful for intestinal injury repair. And I mentioned that one way of potentially avoiding allogeneic transplant to put normal lymphohematopoietic cells into a patient is to correct the genetic defects. And in studies from Paul Orchard, Troy Lund, and Wes Miller, there are ongoing trials of this in inborn errors of metabolism. The stem cells can be taken out. They are corrected in the test tube by putting in viral particles that carry the corrective genes. These genetically modified cells are being put back into patients. And there are ongoing studies here in adrenal leukodystrophy and others planned as well to try to avoid the immune complications of graft-versus-host disease and provide genetic correction. Another novel concept was developed by Jacob Tolar and John Wagner, and this is the treatment of extracellular matrix disorders. So these are disorders in which the cells produce secreted material that helps hold cell types together. And one of these is called collagen 7A. There's a disease known as epidermolysis bullosa. In the epidermolysis bullosa, there's a defect in the adhesion properties between the epidermis and the dermis, so these two are no longer able to stay together. The fibers that do this don't produce the collagen, and the resulting is severe blister formation, not only in the skin, but throughout the gastrointestinal tract. So Jacob did preclinical studies where he took mouse models and was able to correct this with bone marrow infusions. It was the first ability to do that, and through stem cell enrichment processes, he was able to do that in mice and then translate this into humans with John. And this is showing some of the resolution of these blisters. The red is the collagen 7A. It's absent here, virtually absent, and present here. And so this is a brand new therapy, and there are quite a few patients on the ward that are referred here for this process. Now, I wanted to go into the cancer therapy. This is Mark, and I don't know who this is. It was probably before my time. Maybe Mark recalls, but Mark has a special relationship with children, and I think was able to withstand the time periods at which the cure rates for children with cancer were almost zero, and to the point where they're now at least 80% for acute leukemias. And so this perseverance we've all taken on as our challenge. And the goal was to get allogeneic stem cell transplantation to provide a way for tumor reduction by high amounts of chemoradiotherapy and prevent relapse through the immune response. So the chemoradiotherapy is not enough because the disease can come back after transplant. In graft-versus-host disease, which is this donor anti-host immune recognition process where the donor anti-host cells include tumor cells, it can contribute to tumor reduction. It's particularly potent in myeloid leukemias, but severe graft-versus-host disease and its therapy is immune suppressive. So despite a graft-versus-leukemia effect, in addition, tumor recurrence is not uniformly prevented. So the current solutions have included anti-tumor drugs, proteins, and radiation. There are now new drugs, including those that inhibit signaling pathways, radiolabeled antibodies and toxins. There are cell therapies that include natural killer cells that Mike Veneris has developed with Jeff Miller, and chimeric antigen receptor T-cell therapies that I'll mention. There are ways to stimulate the endogenous immune system or the patient's immune system, or that stimulate anti-tumor responses or block those mechanisms that prevent the T cells from from resisting the tumor. And we need better ways of looking at tumor detection and immune assessment assays. So the future solutions will include, I think, more effective T-cell therapies. How do we target cancer stem cells, which we're still defining, or multiple cancer antigens, inhibitory pathways and resistance, and how do we get the infused cells that give to last a long period of time through perhaps stem and memory cells? What are the safe ways of doing this? There's been toxicities from some of these therapies. One way of doing this, which is being developed, is to be able to turn on or off the receptors on the T cells that can recognize the cancer cell, as well as the normal host cell, which shares antigens with many cancer cells, as being different and attack it. And then we may come to a point where we can generate these cells from these pluripotent self-renewal stem cells, so we have an off-the-shelf product which is highly able to be manipulated because these cells can be genetically engineered quite readily. So this has received a lot of attention. This is what a chimeric antigen receptor therapy looks like. So T cells have signaling molecules that stimulate the T cells to proliferate and survive. There are antibodies that bind to antigens present on tumor cells, and they also cross-react with normal cells. These two elements can be fused together so that you have the tumor or host recognition antigens such as CD19, which is present on normal and malignant B cells, and you have the signaling part of it, so the T cell can proliferate and become activated and stay alive. There are variations of this that include now multiple signaling domains intracellularly. Mike Veneris and John Wagner have ongoing Novartis-sponsored studies whereby this fraction here recognizes CD19, which is present on virtually all acute lymphocytic leukemias and many lymphoma cells. And patients are being treated here now, at least I think 5 that I'm aware of. Chris Pennell and Mark Osborne are involved, along with myself, in preclinical studies to look at on-off switches for improved safeties, so that when this binds to normal cells, sometimes there's a large release of cytokines, so it can be very inflammatory and toxic and cause high fevers. And we're looking at ways to with collaborators, to be able to shut off this binding process so that the cell no longer expresses this receptor, and the cytokine storm can be abated. In studies led by Mark Osborne, along with Jacob Tolar, one could modify T cells so that they have the ability to have the anti-tumor effects, but you can take away the T cell receptors that would recognize the host as being foreign and cause graft-versus-host disease. And this can be done genetically. What Mark's done here is to take primary T cells and expand them, and then he can genetically manipulate them to get rid of the T cell receptor so they no longer can recognize the host as foreign, and then expand these for therapy. To do this, there are many gene editing approaches. The one that he's used the most is CRISPR. That allows one to cut the DNA and provide a template so that when the DNA template comes here, the cell can be repaired, but it no longer is able to express the T cell receptor. And these are studies that Mark did using a modification of what was in the literature. He was able to achieve almost 90% elimination of the T cell receptor. And he was able to take these cells and then grow them to large numbers even within a week. So this would allow allogeneic cell therapy where the receptor is only against a particular antigen like CD19, and there's no capacity to cause graft-versus-host disease. This is applicable to also cells that would be generated that are universal donor cells, whereby one could give this to anyone from inducible pluripotent stem cells or from isolated T cells from apheresis units. So another approach to this is to stimulate the endogenous immune system or the infused immune system to be better able to kill the leukemia cells. And so there are a series of molecules that essentially are in two families. One is the tumor necrosis receptor family, and one is an immunoglobulin receptor family. And antigen-presenting cells express the ligands for these. The T cells express the receptors for these. When you engage the T cells that receive signals, you get ability to have the T cells function better. They are less likely to die. They're more able to migrate where they need to go. They're more able to activate antigen-presenting cells, which are important to present tumor antigens so the T cells work better. And in studies that we did a long time ago, you could give animals acute myeloid leukemia cells, and if you gave the acute myeloid leukemia cells, the animals rapidly die of leukemia. You can give donors lymphocyte infusions, which we do is delayed lymphocyte infusions in patients. It does have some effect. But if you give this antibody that stimulates the T cell, endogenous T cell to respond, this alone has a large anti-leukemic effect. And there are now many clinical trials with various antibodies that stimulate the endogenous immune system to do this. On the other hand, there are needs for the co-stimulatory molecules to drive T cell expansion. So what we were able to show is that if you block this is stimulating it. On the other hand, if you block this, you can get rid of graft-versus-host disease, but you also cause a loss of anti-leukemia effects. So this paradigm has been taken into looking at very early post-transplant blockade so that you lose the graft-versus-leukemia effect, but you lose the graft-versus-host disease effect at a time of minimal residual disease, but you don't lose the graft-versus-leukemia effect, which can occur later. And in a pilot study of 10 patients, Leslie Keene at Seattle has shown that graft-versus-host disease is quite low. So that these have been translated into clinic. And in a different approach to stimulate the endogenous immune system, Brenda Weigel led these studies. And this is showing that if you take an acute myeloid leukemia cell, you can stimulate the endogenous immune response by giving dendritic cell vaccines that are matured with the CpG material. So CpGs stimulate B cells to make antibodies. They stimulate antigen-presenting cells to provide tumor antigen expression to T cells to cause a T cell response. And they cause pro-inflammatory cytokines to be released that help drive this. So these dendritic cell vaccines have a substantial increase in anti-tumor effect. They replace lipopolysaccharide, which is really not clinically useful. And in the first study of its kind, Brenda, in a neuroblastoma model, was able to combine chemotherapy with CpGs and show a marked increase in survival with neuroblastoma. And this has led to CpG whole cell vaccines followed by autologous transplant by Ron Levy at Stanford, and the second study by Ron using CpGs and radiotherapy for the treatment of recurrent or progressive lymphomas after allotransplant. And another way of trying to accelerate the immune response would be to give IL-2 to try to drive immune responses. This is data from Cam, Hyman, McDonald that shows that if you give regulatory T cells, you can prevent the lethality of graft-versus-host disease in rodents shown here. But if you give these IL-2 complexes to try to stimulate the expansion of regulatory T cells, which are IL-2 dependent, or to stimulate the endogenous immune system, they can accelerate disease. So these protein therapies are tricky. Instead of that, one can try to get rid of the inhibitory molecules that limit the endogenous immune response, as I mentioned earlier. So these are just showing a variety of these inhibitory molecules. When the T cell binds to the kind of receptors on antigen-presenting cell, the T cell is shut off. And this has actually changed therapy in malignancy patients now. We showed in the first studies of this in transplanted animals that if you give tumor cells, the animals die with tumor. This is AML. If you give spleen cells to try to eliminate this, you get later after transplant where graft-versus-host disease is less, you get some shift. But if you give this with a molecule that blocks this off signal, now you have a marked increase in anti-tumor responses. And this is showing data from Martin Sauer, who is a fellow here, that if you give donor lymphocyte infusions later, they don't tend to function very well because they learn to become exhausted or dysfunctional. And if you block the off signal, now you can rescue these and you can get a tremendous anti-leukemic effect without graft-versus-host disease. You just have to wait till the inflammatory response from conditioning regimen dies down. And this has led to several studies. One that's in press is using anti-CTLA-4 to induce remissions in progressive cancers post-transplant based on these studies. And the other one is to use anti-PD-1, which also blocks an off signal for tumor recurrence post-transplant. And there are 31 patients who have received that therapy now with anti-tumor effects. So graft-versus-host disease that I mentioned several times is a very inflammatory process. This is showing the inflammation of the skin, gastrointestinal tract, tongue, hands, and legs, extremely inflammatory. The immune system, in that case, is not functional and tolerant of the host because the donor immune system has recognized the host as being foreign and attacked it. This occurs in 20 to 70% of patients despite current drug regimens. Chronic graft-versus-host disease, which is a later complication after transplant. It is more fibrosis, occurs in 50% of long-term survivors, and it's the most significant cause of morbidity and mortality. We've used anti-T cell antibodies as a community to try to deplete the T cells. The problem with this is that also depletes the anti-infectious properties and anti-tumor properties. There have been drug and antibody-based therapies to try to be more specific to that, including changing of the ability of cells to home to the gut and various other organs. Post-transplant Cytoxin has been used to specifically deplete rapidly dividing cells, which are thought to be mostly alloreactive, and cell therapies, including regulatory T cells that I'll describe. So we've brought 2 drugs into clinic since the end of Mark's time as head. One is rapamycin, which is used relatively frequently. These were the first studies of their kind to show that rapamycin given to an allogeneic recipient can markedly prolong survival compared to the vehicle control. And the other one is tacrolimus or FK506, and this is showing the same thing, that FK506 compared to the vehicle control can markedly improve survival, and these have been translated to routine use in humans. In terms of the rapid recovery in the immune system that's functional and tolerant, talk to you about regulatory T-cell infusions. These can have high potency and specificity. We may be able to direct their homing, and we may be able to control their persistence. We can harness endogenous or exogenous immune regulatory mechanisms by protein or cell infusional therapies, improve the thymus to be able to select out tolerant cells and to eliminate non-tolerant cells. And we can try to induce tolerance in vivo by blocking these on signals for T cells or engage the off signals for T cells. So I've mentioned a little bit regulatory T cells. These are cells that are made in the thymus. They express this transcription factor known as FOXP3. They express an IL-2 receptor. They go into the periphery. Naive cells are also made in the thymus. They go into the periphery, and they can produce pro-inflammatory cytokines, anti-inflammatory cytokines, and very specific cytokines such as IL-17, which causes autoimmune disease, and microbial immunity. They can also be induced, naive cells can also, under very particular conditions, be induced to become suppressor cells in the periphery. So the balance between regulatory T cells and effector cells is critical. If you have a lot of effector cells, you get autoimmunity, you can get graft rejection, and this is useful for cancer therapy, but is detrimental to these other processes. And this is under high inflammation, it favors effector cells. Under low inflammation, it can favor regulatory T cells, which have been useful to treat autoimmunity and prevent graft rejection. So these are studies that Pat in the lab, and she noted that if you give T cells and now you combine those with regulatory or suppressor T cells, you can have this tremendous protection against graft-versus-host disease. This was the first report of this type of approach, and this has been seen by every lab that has tested this therapy. These are extremely potent cells that have not had any variation between labs and different strains of mice. So Wayne Godfrey, when he was here, then brought this into the clinic, and this is this population in humans. It expresses CD4 and the IL-2 receptor. And in peripheral blood, it's relatively non-discrete. It has these high cells are the suppressor cells, and these lower cells are not suppressor cells. And using a CliniMax device where you could isolate the CD25 positive cells, in the early days, we were able to get isolation of CD25 positive suppressor cells, but there were also a lot of other cells that came with it that were not suppressive. So John was doing cord blood here, and we reasoned at the time that cord blood might be a good source because first, it causes less graft-versus-host disease, and second, the IL-2 receptor positive cells that are typically activated in response to pathogens or food allergens, et cetera, are not going to be present as much in utero. And you can see this is a much more discrete population. And using a trick to isolate the highest IL-2 receptor positive cells, now you can get a much higher proportion of these suppressor cells. And this was taken to the clinic by Claudio Brunstein and John Wagner. And this was the first of its kind using completing a full phase 1 study of ex vivo expanded cord blood cells. So this is giving adult patients with hematological malignancies 2 cord bloods as a source of stem cells, taking a third cord blood unit that was 4 to 6 out of 6 matched with the recipient, not taking into account the donor grafts, and expanding these first. And in comparison to 108 historical control patients which had a 60% incidence of acute graft-versus-host disease grade 2 to 4 on a 0 to 4 scale requiring systemic steroids, this rate is high because there are 2 cord blood units and the T cell dose is twice as high, but you needed 2 cord blood units to fully reconstitute an adult because the reconstitution is dependent on how many stem cells you give. Despite the fact this was a phase 1 dose escalation curve, there was a significant reduction in graft-versus-host disease from 60% to 43%. Viral and fungal infections did not go up, in fact, were lower, and there was no increase in relapse, which was always the concern. These were not powered for any of these analyses. These conclusions are not firm, but it did spur us on to do more studies. Now, one of the problems with these studies is cord blood ranges from 50 to 250 cc's or so. It contains not that many cells in total. After purification, there were 6.5 million cells isolated. This could be expanded to 3.8 billion. But 22% of the patients didn't have sufficient expansion. In addition, the patients that did, the average number of regulatory T cells to effector cells was 1 to about 6. In rodents, that optimal ratio is 1 to 1. If you hit 1 to 2, you start to lose some of the biological effects. So Kelly Hippen developed an approach to try to optimize expansion of these cells. Everybody in the field had been running at about 200-fold expansion or so, up to 400-fold. But we reason here that one of the problems is that the regulatory T cells, which are stimulated through with anti-CD3 and IL-2, amongst other things, they were re-stimulated by almost everybody, by everybody, at day 7, because it's a very convenient time point. However, these regulatory T cells are very large at that point. And unfortunately, cells that are large, when you try to stimulate them, either don't become stimulated or they die. They don't want to be re-stimulated when they're so large. So using a formal cell sizer measurement, we waited until they hit their lowest cell size and then re-stimulated them. And these are showing 5 stimulations over a period of 70 days. So what that allowed is this is what you get from the fold expansion from the 1st, 2nd, 3rd, 4th, and 5th. So now you have this incremental ability to tremendously expand the number of regulatory T cells, which frankly had been the rate-limiting factor into bringing this therapy into the clinic. And these cells were still able to suppress an immune response. This is at 1 regulatory T cell to 4 effector cells. And this is retained pretty well through 4 stimulations with some loss by the 5th stimulation. So this led to a study that Claudio and John ran to go from the highest dose in the first trial, which was 3 million cells per kilo, with a planned escalation to 300 million cells per kilo. It was stopped at 100 million cells per kilo because that was a cost-effective approach that gave us biological efficacy. 14 patients were enrolled, 11 were treated, 2 had technical issues not related to the regulatory T cells, but a change in how they were being grown. 1 patient withdrew consent. So instead of having 200 to 400-fold, now this is 13,500-fold after 1 re-stimulation. And these are the data with 23 patients that received the GVHD prophylactic drugs alone. This is the rapamycin and mycophenolate versus 11 patients that received regulatory T cells. This patient is scored as positive. I would say it's equivocal. It's scored as positive to be conservative. The biopsy was inconclusive. This patient received 3 weeks of steroids, too short for graft-versus-host disease treatment in general, too long for cytokine engraftment syndrome, which is a cytokine effect. We don't really know. Nonetheless, there's a significant reduction of grade 2 to 4 GVHD and almost a significant reduction of the most severe graft-versus-host disease, where none of the patients received that and developed that in the first 100 days post-transplant. So a second approach to this, as I mentioned, is to try to not isolate this rare population of 2 or 3% of cells, 1 to 3% of cells in the blood, but to make the cells that are very abundant become suppressor cells. And as I mentioned earlier, the naive cells under certain conditions, here I'm showing tumor transforming growth factor beta receptor, transforming growth factor beta and rapamycin can drive these to acquire suppressive cell properties. So these cells are easy to isolate. They're very high in initial numbers. They can expand at least 1,000-fold. You can produce 250 billion of cells in 12 days. And they have distinct homing mechanisms that are different than thymic-derived regulatory T cells, so they may be able to be used together. So this is showing one experiment from Kelly. This is the thymus-derived cells. In this experiment, he had 850-fold expansion. Suppression is relatively similar. And in vivo, in terms of inhibiting human peripheral blood mononuclear cells induced GVHD in immunodeficient mice at a suboptimal ratio of 1 of those cells to 3 of the effector cells, these were relatively comparable. And this led to a clinical trial that Margie's leading along with John that's ongoing, which is to give these cells in a dose escalation fashion up to doses of 10 to the 9th per kilo with cyclosporin. And we'll see what happens at the end of this, but this is a first-in-human study of induced regulatory T cells in any patient population. So I mentioned chronic graft-versus-host disease is a fibroproliferative process. This is showing some pictures of this. Instead of the angry inflammation, there's a lot of fibrosis, there's joint contractures, there's ulceration. It involves most of the epithelial cell surfaces in the body, and it is very distinguishable from acute graft-versus-host disease. I mentioned it's 50% of transplant recipients who survive 6 months, major source of morbidity and mortality in those who don't relapse. It's fibroproliferative. Antibody production, which is antibodies that can recognize host tissues as foreign, have been seen in many studies, not all. And in the tissues, there's very few cells present sometimes. They're mostly fibrosis. So the challenges in this field, which have led to very few new agents in the past several decades, is the standard criteria for diagnosis, as well as choosing and evaluating therapies have been less than clear. Preclinical models that had been available before we started to tackle this problem had questionable relevance. They were dominantly non-irradiated recipients, which has no relationship to the human. They developed lupus-like syndromes, particularly involving the kidney, or they had skin disease alone, which chronic graft-versus-host disease is not often skin disease alone. So Angela Panascolis and Mortari developed this model, Cytoxin and irradiation, and the trick was to give low doses of T cells. So the low doses of T cells didn't cause animals to lose a lot of weight. The animals survived long term. And these low doses of T cells were chronically stimulated so that they would have activation for a period of months. And then one can look at then treatment after establishment of disease, which takes a month. And so she and individuals in my lab as well described involvement of the lung, the liver, the tongue, spleen, thymus, and intestine of this. So this is a true multi-organ system disease, which had generally been absent in the field. And she noted in the original publication that there was a significant involvement of the lung. This is showing infiltration around one of the bronchioles that simulates bronchiolitis obliterans syndrome. That is a graft rejection process that happens in lung transplant, but here you can see the air entry and exchange would be very minimal. If you develop this, which happens in up to 14% of chronic GVHD patients, there's a 65% 3-year mortality, and frankly, the only true therapy for this is lung transplant. So in this model, one can look at fibrosis. And this is showing chronic graft-versus-host disease mice, and this is fibrosis. This is a stain. You can see there's no air that's going to get through this bronchiole. It happens in the liver too. One can quantify this as going much higher in chronic graft-versus-host disease mice compared to no chronic graft-versus-host disease mice in the lung and the liver. The same is seen in the intestine. These mice can be intubated and mechanically ventilated under anesthesia. And when you do that, this is day 28, you can have high resistance because the lungs are stiff. You can have high elastance, which is loss of recall properties because the elastic properties have been damaged. And you could have low compliance, which is resistance against pressure. And in studies that were done between my lab and Angela's, we found that there were immunoglobulin proteins that were deposited in the lung. This is IgG2C because the donor strain happens to dominantly make IgG2C. Here's it showing in the lung. It's the same in the liver. And this is then simulating the patients that have antibody production in humans. And so turning to what causes antibody production, I won't take you through the details of this, but suffice to say that naive T cells can enter lymphoid organs where they can collaborate with B cells in these organs. And these B cells make plasma cells that produce copious amounts of antibody. The regulatory T cells that I mentioned can collaborate, can home into these same organs, and they try to suppress this response. So the hypothesis then was it was these germinal centers or these lymphoid follicles that were the source of these antibodies, and it wasn't antibodies being produced in the tissues, but antibodies being produced elsewhere. And in these studies, looking at no chronic graft-versus-host disease versus chronic graft-versus-host disease. This is staining for 3 antigens, and together this is what the germinal center looks like. And you can see they are much larger here. They have they're higher in number. They're bigger in size in chronic graft-versus-host disease. And if you give reagents that block this germinal center production, you can treat the disease. So the other part of doing that, we were going to bring that to clinic, except Biogen and IDEC closed that program, and that was a lymphotoxin beta receptor immunoglobulin. So another approach to doing this is to try to stimulate regulatory T cells to suppress the disease. These are data from the Dana-Farber, and what they show is that there are very few regulatory T cells present in peripheral blood of chronic GVHD patients at baseline. This is baseline. If you give low doses of IL-2, you can partially ameliorate the symptoms of that, and that's because this population expands rather dramatically. And this is shown here, the regulatory T cells compared to the conventional T cells. That ratio goes much higher during IL-2 administration. You stop the IL-2, it goes down. In studies that I've been fortunate enough to collaborate with the Dana-Farber group, it's shown that the regulatory T cells aren't made very many in patients because there's thymic injury, and the cells that are made tend to die. They don't have the anti-apoptotic or anti-death pathway proteins that allow them to survive, and the IL-2 helps with that. So going back to this model, Cam Hyman McDonald, who's an MD PhD student here, and Ryan Flynn, who's a postdoc, supported by CCRF to do these studies, compared wild type versus a knockout regulatory T cell where the regulatory T cells could not home to the germinal center to suppress this response. And you could look at, given these cells day 0 or day 28 as therapy. This is for prevention, this is for therapy. And I showed you before how IL-2 complexes could be bad for acute graft-versus-host disease because it could make it worse. But in chronic graft-versus-host disease, at least in this model, here's showing the pulmonary dysfunction of chronic graft-versus-host disease. If you give these complexes as treatment, you can now reverse the disease. And why is that? That's because there's very low inflammation here compared to acute graft-versus-host disease. And the regulatory T cells have the highest level of the IL-2 receptor, so they tend to divide preferentially over the effector cells. And if you look at regulatory T cell infusional therapy, you can give the cells on day 0, 28, and you reverse the disease or prevent or reverse the disease. You can take a regulatory T cell that doesn't go to the germinal center and it will still prevent the disease because there are likely not germinal centers there. But if you give it as a therapy, if the regulatory T cells can't get to the germinal center, they don't work very well. And so this has a lot of implications for the field, and particularly combining IL-2 complexes with regulatory T cell infusions, but also the fact that the regulatory T cell infusions may be preferred over the proteins because there are patients with chronic graft-versus-host disease that have inflammatory components and they may not do well. And I've shown that already. So in summary, I've tried to go through some of the advances and challenges that we faced in the field. Pat Taylor led many of these studies, and she's retiring after 28 and a half years with me. We've collaborated with Angela Panaskolsis-Mortardi and Mathenge and Ryan Flynn and Cam Hyman McDonald did the regulatory T cells and chronic GVHD models, Michelle Smith on the T progenitor cells. Tiffany and Bo were PhD students in the lab. Many present and former lab members contributed to this, and we've been very fortunate for collaborators with Kelly, Jacob, Mark, Heather, Troy, Mike, Veneris, Mike, Kaiba, and Chris Pinnell. The clinical studies from John, Margie, Mike, Heather, Troy, Paul, Wes, Angie, and Kristen Evans. This is a team approach. You cannot do these studies without a team approach. We all have to work together, and we all have different roles in this. And I think that's why the division has been so successful, is Mark has created an environment that fosters teamwork, collaborations with Brenda and Bruce Bostrom, with the adult transplant program, only some of those I've mentioned, the GMP molecular and cellular therapy facility that allows cell infusions, and a great list of mentors, Mark, Norma, John, Harry Orr, whose lab I was in for a few years, and Dan Valera, and local support, particularly Children's Cancer Research Fund, as well as the division, department, families, patients, faculty and staff, my own family, and national funding agencies. Thank you for your attention.
Thank you, Bruce, for an amazing tour de force of translational medicine and also a real testament to the history and legacy that goes before all of us of what sets the path and how we build on those learnings and how we can translate what seemed like simple observations years ago to now as standard of care in the field. And it's a pretty amazing testament to the work that has started here and continues here and paving the way. And thank you to Mark for really setting the stage for all of this to happen. And Norma and all of the others that went before us to pave the way and work together as a team, because this really is a huge collaborative effort. So with that, I'll open it up if there's any comments or questions for a few minutes.
A bit over time, so that's...
Okay. Joe.
Yeah.
So just a question about the using regulatory T cells outside of the transplant setting. Rheumatoid diseases, chronic autoimmune disease. Any experience or pathway there?
So the FDA required us to complete our phase 1 study before they would let them be used for any other purpose. That was then taken into diabetes, type 1 diabetes by Jeff Bluestone. He's reported that on this that it has some effect to reduce the loss of C-peptide. There are multinational studies now in kidney transplant, giving one of 9 different regulatory populations to patients to try to prevent kidney graft rejection. There's another one starting in liver graft rejection. There are trials being looked at for colitis and for uveitis.
Anyone else?
Thank you.
Margie?
Yeah. Okay.
You had fabulous ideas of future plans and projects to move the field forward, which I assume many of them, you want to do or see done in the next 10 years. Move ahead to 50 years. Where do you think transplant's going to be? Is there still going to be a role for allotransplant, or is there just going to be engineered cells that are administered very much in a tailored fashion for a patient?
I think it has to go eventually to off-the-shelf products. I just don't see this as being something that you want to continue using bone marrow harvest, peripheral blood. So whether those will be from pluripotent stem cells, whether those will be from ex vivo manipulated cell populations, I don't know. But the off-the-shelf products allows you to have large numbers of cells that you can use as universal donors for particular purposes. Whether that's all going to be necessary or not, for some things like extracellular matrix diseases, it's hard to imagine you can get the correction that you need systemically, or for some of the storage diseases. For some of the cancers, we have things to solve yet, and not every cancer stem cell is going to be known, and not every one of those is going to have a good target that doesn't cross-react with the heart, the brain, or others. So I think that's an unknown. Thank you.
I think with that and the prediction for the future, I'd like to thank everyone for being here and celebrating with us the 25th anniversary Nesbit Lectureship. Thank you all.